So today we're going to look at MCAT Immunology. So the question is whether or not you've had immunology formally in a class, seen it in biology, physiology, or some other place. So the MCAT definitely has immunology. Here's the actual AAMC MCAT Content Outline. It's fairly comprehensive. So we see the structure and function of proteins of the immune system. Of course, they must be in the antibody and possibly MHC, as we'll talk about. There's also in category 3B organ systems, you have a number of things, including innate versus adaptive immunity, T and B cells, macrophages, phagocytes, and you can see the rest of the list there. We're going to go over all these things. They're pretty important. We'll look at the most basic features of them, so it'll be incredibly introductory, but we should also appreciate that antibodies are used in lab techniques. Molecular biologists and biochemists use antibodies for ELISAs and Western blottings, as well as flow cytometry and other techniques like CHIP all the time. We have passages and questions on our QBank that detail all these things. So where do immune cells come from? Well, in adults, they come from hematopoiesis. So it creates immune cells. So what you have is a multipotent hematopoietic stem cell, and this gives rise to myeloid cells or lymphoid progenitors. On the right, what you see is that you can develop natural killer cells, NK cells. You don't have to know anything about these for the actual MCAT. You shouldn't confuse them with T killer cells either. So the small lymphocytes give rise to T cells and B cells of lymphoid origin. T cells express the T cell receptor, as you see here in green. B cells express the B cell receptor, which is basically an antibody that is trapped in the membrane. And then B cells can further differentiate and give rise to plasma cells, which actively secrete B cells. Over here on the left, we have common myeloid progenitor giving rise to megakaryocytes and thrombocytes. You probably call them platelets. And then we see mast cells, which are involved in allergic responses. Erythrocytes, of course, deliver oxygen. And then the myoblasts give rise to various types of white blood cells, including monocytes and macrophages. We'll see a little bit more about macrophages. These are actually antigen-presenting cells. They can engulf and take in by phagocytosis, bacteria, as well as viruses, chop them up, and then present their protein sequences to the MHC complex, and that activates T cells. So then innate versus adaptive. So innate is what you're born with. It's the physical barriers. Your skin is innate, mucous membranes, the pH of your stomach. And then there's inflammatory mediators like complement, cytokines, and prostaglandins. The MCAT AAMC Content Outline doesn't really talk much about complement. You'll see it in medical school. It's pretty important. Cytokines are going to be small peptides that are secreted by a variety of cells that are going to induce signaling pathways into their downstream effector cells. Prostaglandins are involved in inflammation. They're generated from arachidonic acid. These are incredibly important. A lot of our drugs, like COX inhibitors, are designed to actually reduce or completely inhibit the synthesis of prostaglandins. And then we have cellular components. These are phagocytes, or neutrophils, eosinophils, basophils, and mast cells. I mentioned those before. We're going to look at macrophages. These are actually antigen-presenting cells along with dendritic cells. So the macrophages will be incredibly important, derived from monocytes. So acquired immunity. This is composed of B and T cells. And so this is activated by the innate immunity system. So innate can either get rid of stuff, your skin, for example, or what you do is you take up something by phagocytosis and you present what you took up to the actual B or T cells. So as I mentioned, B cells express B cell receptor and make antibodies. T cells are the ringleaders of the immune system. They mature in the thymus, express the T cell receptor, and they're activated by antigen-presenting cells such as macrophages or dendritic cells. So the major lymphoid organs. So we know that in adults, hematopoiesis is in the bone marrow, but in the fetus, the actual liver is an immune organism, and it produces lymphoid cells in the beginning. But once you're born and you start to develop, then the bone marrow takes over. So there's two primary sites, and that's the bone marrow for, as I mentioned, hematopoietic production of myeloid and lymphoid cells. And the thymus actually receives the bone marrow T cells. And this is where the action takes place where you decide if you're self or non-self, because one of the major functions of the immune system is to determine self versus non-self. You don't want to react with yourself. When you do, it's called autoimmune, and we'll see a passage on autoimmunity and Graves and Hashimoto's disease a little bit later on. So the lymph nodes and spleen are secondary sites of their secondary lymphoid organs, and this is where you actually clear things away and where you activate antigens, or antigens activate lymphocytes, I should say. So let's take a look at macrophages. They're called sentinel cells. They're going to receive the actual information. There's an invader here. So imagine, for the sake of argument, you've eaten some bad chicken, you got salmonella poisoning, and so it's in your GI tract, and what you have are these M cells, these microfold cells that exist along with the other types of things like goblet cells in your GI tract. And what they have are receptors, and we'll take a look at the receptors in a moment. They're called toll-like receptors and other things as well, but toll-like is pretty important. And what happens is that you can bring these in by endocytosis and then ultimately deliver it to the lamina propria, which is enriched in basically lymphoid tissue. This is a so-called Peyer's patch. And here, resident macrophages are actually going to recognize these things because they can recognize the flagella or other parts of the bacteria in what's called pattern recognition. And they take it in and present this actually to the T cells and the B cells, and then what you have is an immune response that's generated. So then the pattern recognition can occur from several things. For example, there's lipopolysaccharide, or LPS. It's expressed on gram-negative bacteria, as I'll show you in a moment. And what this basically is a bunch of antigen repeats here. These are basically sugars attached to a lipid here, and you can see some of the glucosamine monomers right here. So this is LPS is composed of lipid A plus O antigen repeats. And so what we see is that LPS is on the surface of gram-negative. So E. coli, salmonella would be gram-negative, but not on the surface of gram-positive, and that would be something like a bacillus strain. So the difference between gram-negative and gram-positive is the expression of LPS, but it's also the cell wall and the outer membrane. So gram-negative have an outer membrane, gram-positive don't, and the cell wall is thick in gram-positive, but not in gram-negative. And you can see a better representation of it right here. So here's the LPS that's stuck on the outer membrane here. And basically, this is something that the immune system is going to recognize as a feature that's common to invaders like bacteria. So notice that the peptidoglycan layer is pretty thin in the gram-negative, but it's super thick in the gram-positive. But the gram-positive doesn't have an outer membrane. And so another pattern feature that's recognized is the actual flagella of bacteria. These are things that are involved in bacterial motility. And you can think of it as basically like a mini machine that's designed to help the bacteria swim towards attractants or swim away from repellents or dangerous chemicals that might hurt it. So here what you have is this filament is composed of monomers of a protein called flagellin, has a hook. And so basically it's a rotary motor. Now, bacterial chemotaxis is specifically listed on the AAMC Content Outline, and it's quite important because it's used by organisms to actually help invade, to swim towards things. And so what normally happens is that bacteria just are randomly walking. They go one way and another, and they never really get anywhere in the net scheme of things. But this is called a random walk. But in the presence of an attractant like sugar, for example, this random walk becomes biased. And the way you perform this is by having your flagella bundle. So if the flagella are all moving counterclockwise, they tend to bundle, and the bacteria can actually swim and make net progress. But for example, if you're going to be exposed to, say, chemicals that can hurt you, then you're not going to want to swim towards them anymore. They're repellents, not attractants. And then the flagella actually rotate the opposite direction clockwise. And when you do that, they're all discombobulated. And as a consequence, what you do is you tumble away from the actual repellent. So here's the toll-like receptor. They're pretty common. And so toll-like receptors recognize a number of things. They can recognize bacteria, viruses, even nucleic acid. So you might think, well, nucleic acid, isn't that self? Well, in the extracellular milieu, that it's not necessarily self, because you can have degraded bacteria or viruses that have shed their outer protein coats, and so it can be recognized potentially as something that is foreign. And so what happens is you can see that no matter how or what you recognize by the toll-like receptor, whether it's a virion, a virus, bacteria, nucleic acid, etc., then what happens is that you generate these signaling pathways that activate a number of things. I don't think the signaling pathway per se is important to remember, although NF-kappa B is a major, major player in the immune system. It's probably going to be on the MCAT sometime. And what happens, though, is that you ultimately generate cytokines. This is an interleukin right here. And so this thing called the inflammasome is actually going to be involved in the process of generating various molecules that will signal the immune system to actually perform. This could be activation of T cells, this could be B cells. So here's phagocytosis of, say, a macrophage. So the bacteria that's in the lamina propria hits a macrophage, and here's your toll-like receptor here. It gets engulfed in the phagosome, and then this merges with the lysosome, which we know is a really acidic organelle. And so what's going to happen then is that you're going to hydrolyze and just chew up the foreign invader. And what you do is you actually save some of the antigen peptides over here, and you put them onto a protein called major histocompatibility complex, MHC. Now, MHC is made by the host. It's translated off of ribosomes. It goes through the secretory pathway where it's going to be ultimately secreted on the surface. And this MHC can actually bind peptides, and it presents them to T cells. It says, look, this is what the cat brought in. It brought in a bacteria, and here's a piece of it, and you should know that because you need to do something about that and make an immune response. And so here's what MHC looks like. There's two different types. I don't know how important that is to remember. It depends on the type of T cell. You should know what T cells are. There's many types of T cells that MCAT probably won't make you discriminate on the types. It could give you a passage that gives you information about that. But so what you have, whether you're in type 1 or type 2, what you basically have is this protein forms a groove with the alpha 1 and alpha 2 subunits or the alpha 1 beta 1 subunit, and you can actually present this on the surface of a cell. So we switch over to the clonal selection theory of adaptive immunity. And so we know that B cells express B cell receptors on their surface. And in fact, these receptors can bind a cognate antigen that's specifically designed like a hand in a glove, an enzyme substrate type of interaction. So we get really tight binding in some cases. And so there's numerous B cells expressed, each one's expressed its own unique B cell receptor. The B cell receptor is basically the antibody that recognizes the antigen, but the antibody stuck in the membrane. We'll see later that it can actually be secreted. So the B cell receptors that recognize the self have already all been selected against, okay, under normal circumstances. And so what these things are doing is they're circulating in various tissues, they're in lymph nodes, et cetera, and they're in G0, they're quiescent. But when it encounters, one of them encounters its cognate antigen, which could be something floating around from a virus, what happens is that you do a signaling response that actually you endocytose the receptor into the cell and you start degrading it through the lysosomes. And then you present a piece of the peptide from the antigen, which perhaps is a virus or a salmonella, on the MHC. Okay, and then what MHC does, it finds its T cell, its cognate T cell receptor that could recognize that peptide antigen, and the T cell starts making a bunch of cytokines. It secretes cytokines that are recognized by the B cell, and then what happens is that the B cell is going to induce all kinds of gene expression programs that are going to actually secrete the antibody. And now the antibody when it's secreted, no longer has the capacity to dwell inside of the membrane because the exon that encodes for the piece of the protein that keeps it in the membrane has been skipped by alternative splicing. And what you do then is you secrete a whole ton of antibodies here, and you start expanding, you start proliferating this one specific B cell population. And ultimately, these B cells will turn into plasma cells where they're going to secrete lots and lots of antibodies. Now, antibody structure and function is a really critical thing to know, not only as a doctor, but scientists too. They're used all the time. So let's take a look. This is the classic Porter and Edelman structure of the antibody. What it is, it's composed of 2 heavy chains and 2 light chains in such a manner that what you have are the variable regions over here. The variable regions have a light chain component as well as some of the heavy chain component. The heavy chain is heavy because it's larger. It's a larger protein than the light chain, which is smaller. And so what you see then is that these subunits are held together by disulfide bridges represented in green. So then it's a secreted molecule that has disulfide bridges. And over here, you see there's 2 parts. There's FAB, the fragment that binds the antigen binding. And there's also FC, fragment crystallizable. That's historically named from the Porter-Edelman studies. But the important thing is that the FC contains the constant region, okay? So you have variable region, constant region. The variable region here contains the antigen binding sites. Notice that each antibody has 2 antigen binding sites. And what you can do is you can actually reduce these disulfide linkages with agents such as dithiothriitol or beta-mercaptoethanol and dissociate the antibody into its constituent chains. And what happens in diseases like multiple myeloma, and we have a passage on multiple myeloma that's going to challenge your ability to understand antibody structure and function. What happens in multiple myeloma is a disease of proliferating plasma cells. So your body's making too much antibody. And when it does that, it actually makes excessive amounts of light chains, for example, here. And these light chains, if they don't have an actual heavy chain to bind to, if it's not properly stoichiometrically regulated, these free light chains build up, and they can do a number of things. They can actually hurt your kidneys and cause all kinds of things. So in multiple myeloma, what people measure is the actual free amount of light chain. But as you can imagine, since you also have light chains that are bound to heavy chains, measuring the free amount of light chain is actually a difficult task. It took years to develop specific reagents that can help you do that. So classes of antibodies. So we know that the constant region determines the class of antibody here, and IgM is the first one that's made. It functions in the primary immune response. It's a pentamer, and it binds the B cell receptor. But what you can do is you can switch over. You can isotype switch the IgG, which is the major immunoglobulin. It crosses the placenta, but the other ones are located in different parts of the body. IgA is in the mucus. IgE binds to mast cells. We know it's mediated in the allergic response. It releases a lot of histamines. That's why people take antihistamines like Benadryl. And IgD is co-expressed with IgM on B cells. So how do you build an antibody? This slide looks potentially complicated, but I'm going to take you through it step by step. So what you need to do in order to build this antibody molecule here in the bottom right, what you do is what's called VDJ recombination and isotype switching, which is also called class switch recombination. So to build an antibody molecule, so in the old days, people knew there were lots and lots of antibodies, and there were millions of different types of antibodies. And they said, well, if it's one gene and one enzyme, or one gene, one protein, according to the classical biochemist Beadle and Tate theory, then would you need millions and millions of genes to make all those antibodies? And if you did, how could the chromosomes possibly be endowed with all that information? So people were skeptical. So then guys like Tonegawa figured out things like VDJ and isotype switching later on. So what you have is a bunch of exons called V, D, and J. And these exons are going to encode for the variable region, but downstream you have constant region exons. And by splicing and dicing, you can put V2, D1, J3, or you can have any permutation. And what that makes is a unique variable chain here. And there's lots and lots of combinations. So the splicing and dicing of various combinations in VDJ explain, at least in part, how you make such a diverse repertoire of antibody function. And so the first one, here's the variable region here. The first constant region that gets expressed is the mu, which is the M for IgM. Okay, and so you make an IgM molecule initially, as I mentioned. But then what can happen in response to cytokine signaling from T cells, et cetera, is that you can initiate a second DNA break program where you actually cut out this region here. So this boxed region right here gets cut out. And as a consequence, what you do then is you lose this information and you juxtapose the gamma 1 sequence next to the actual variable region over here. And what you're seeing then is that you're putting this light green gamma 1 in position here. And what you do is you switch from an IgM to an IgG, and then you get rid of the intervening sequence. And by juxtaposing these regions together, what you can do is you can isotype switch, but notice that the IgM that was made and the IgG that was derived from it from isotype switching only changes the constant region. It doesn't change the variable region. So antibody isotypes bind the same antigen. They have different constant regions because what they do is they work in different parts of the body. So one of the problems with creating DNA breaks at the immunoglobulin locus is that if you don't properly fix these breaks, there's problems that can generate translocations that are causal for numerous diseases. So we find that about 80% of all known lymphomas have some sort of DNA break translocation problem. And one of them is called Burkitt lymphoma. And what happens at the immunoglobulin H locus that breaks with either VDJ or class switch recombination, if it doesn't repair that break normally, then what happens is that DNA broken end can invade other chromosomes. And one of the chromosomes is notoriously invades contains the c-MYC gene. Now, normally c-MYC isn't expressed very much at all, but when you juxtapose this gene under the control of the immunoglobulin H promoter, what you do is you start cranking out more c-MYC than normal. And given the fact that c-MYC is a transcription factor that regulates 20 to 30% of the genes in the chromosomes, all chromosomes, it's pretty busy. What you do is you generate perturbed proliferation, you get Burkitt lymphoma. So we've discussed some antibody techniques before, but we can never talk about immunology without talking about lab techniques. The most common one is the Western blot. And basically this is designed to recognize proteins in any given sample. So what you do is you run an SDS page gel. You can run it under reducing conditions or under native conditions. And if you run under native conditions, then protein complexes should be detected as a whole entity. You can transfer to a membrane, and then you block with a non-specific protein and add an antibody that's designed to detect the protein of interest. It's called a primary antibody, or alpha. And it recognizes this protein here that was in the extract. It doesn't recognize it apparently in lane 2. We'll talk about that in a moment. And then what you do is you wash away, because these antibodies can non-specifically bind to other epitopes. And then you add a secondary antibody that's been decorated with a fluor, some detectable thing. In some cases, people use radioactivity. Other times they use chemiluminescent fluors. Then you wash away complexes that don't bind specifically. And then what you do is you have your actual detected Western blot band. There's nothing in lane 2. So this raises the issue of controls. You always have to do controls in all experiments and in Western blots specifically. So a negative control would be a sample that's known to not have the antigen of interest. It could be a genetically knocked out cell line, for example. Now, a negative control should never give you any kind of response. A positive control, on the other hand, would be a cell line or a recombinant protein that is actually specific for the antibody or contains the protein that the antibody is supposed to recognize. And you can also do peptide competition assays. So imagine that this antibody was raised against a peptide. It can, if you add a bunch of peptides in here, it should bind to the peptides and swamp out the capacity to bind the epitope that's actually on or derived from the cell extract. So always think about control experiments. So there's also ELISA assays. We're going to look at a couple of them. Here's the indirect ELISA. This is used to determine the concentration of an antibody in solution. So what you do is you take a sample, it could be a blood sample or even a urine sample or some biological sample, and you coat these plates. These plates have there's a number of things that you can be used to coat them. They're hydrophilic in nature, and so we're expecting our antigen to be sticky through electrostatic interactions and stick to the plate. Then what we do is we add the antibody to it, okay? And this antibody can come from another biological sample. For example, did you make antibodies against coronavirus? The antigen could be derived from a sample. It could also be derived from in vitro purified spike protein for the actual detection of the antibody in solution. And so what you do is you add your antibody. If it's there, it's going to bind to the antigen. Then you add your secondary antibody that's labeled with a fluor. So in a sense, it's analogous to the Western blot. And if you detect, then the argument is that you actually have made the antibody against the antigen of interest. Of course, you got to run controls too. What would those look like? Well, one control, of course, would be to run a blank plate where there's actually no coating with the antigen. You should theoretically get no signal whatsoever. So that's the indirect ELISA assay. So we also go to the sandwich ELISA assay. This measures the concentration of antigen in the sample. And so here what we do is instead of coating with an antigen, we coat the plate with a capture antibody. And then after you add your sample that contains the antigen of interest here, what you do then is you actually make an antibody antigen complex, and that is going to hold the capture antibody and the antigen together, and then you add your sample, and then this is going to actually form, when you add your secondary antibody sample, what you do is you form a sandwich, as the name implies. And so the idea is that the second antibody recognizes the antigen, but in a different place. And so we have indirect ELISA assays designed to measure the concentration of antibody in a sample, and we have sandwich ELISA assays designed to measure the concentration of an antigen in a sample. It uses the notion of a capture antibody and a second antibody that recognizes the antigen at a separate epitope. And with all this in mind, you can take a look at the comparison right here, side by side. You should definitely know this for the MCAT. And with that in mind, let's do some challenging workshop passages that are going to go over our ability to apply this information.